The capillaries permit the exchange of nutrients and wastes between the blood and tissue cells. Capillary exchange occurs via three basic mechanisms: diffusion, transcytosis, and bulk flow. The most important method of capillary exchange is diffusion. Oxygen, carbon dioxide, steroid hormones, and other lipid-soluble substances pass through endothelial cell membranes. Glucose, amino acids, and other water-soluble substances diffuse through intercellular clefts and fenestrations. Transcytosis is used to move small amounts of large lipid-insoluble molecules, such as large proteins, across capillary membranes. In this process, substances contained in vesicles enter endothelial cells by endocytosis and exit by exocytosis. Bulk flow is the movement of large volumes of ions, molecules, or particles between blood and interstitial fluid. The movement occurs when a pressure gradient pushes water and dissolved solutes out of or into the capillary. Bulk flow is always from an area of high pressure to an area of lower pressure, and continues as long as pressure differences exist. Substances in the flow move in unison across fenestrations and intercellular clefts. Fenestrated capillaries allow more bulk flow than continuous capillaries. Tight junctions at the intercellular clefts of continuous capillaries allow little bulk flow. Most solutes can cross with water. However, most larger proteins and formed elements in the blood, such as red blood cells, cannot pass through the fenestrations of the endothelial cell. Bulk flow plays an important role in regulating the relative volumes of blood and interstitial fluid. It does this by balancing specific pressures. Bulk flow is given two names, depending on the direction of movement. There are four factors that determine the net direction of capillary exchange. Blood hydrostatic pressure, generated by the pumping of the heart, promotes filtration by pushing fluid out of the capillary. Blood colloid osmotic pressure is caused by the colloidal suspension of plasma proteins that are too large to pass through fenestrations or gaps between endothelial cells. This pressure acts as if it pulls fluid into the capillary and promotes reabsorption. Blood hydrostatic pressure combines with interstitial fluid osmotic pressure, the slight pull of fluids towards the interstitium, to promote filtration. In opposition, blood colloid osmotic pressure and interstitial fluid hydrostatic pressure, which is near zero under normal conditions, promote reabsorption. The balance of the filtration and reabsorption forces, called net filtration pressure, determines whether blood volume remains steady or changes. At the arterial end of the capillary, filtration pressures are stronger by 10 millimeters of mercury. At the venous end, reabsorption pressures are stronger by 9 millimeters of mercury. As these typical pressures illustrate, about 90% of the fluid volume that is filtered out of the capillary is reabsorbed. The remainder drains into lymphatic capillaries to form lymph. An abnormal buildup of interstitial fluid results in swelling, known as edema. Edema is a symptom of increased blood hydrostatic pressure, or capillary permeability, decreased blood colloid osmotic pressure, or a problem with normal lymphatic drainage. Blood moves through vessels at a rate that is inversely related to total cross-sectional area. For example, capillaries, when all of them are added together, have a higher cross-sectional area than arteries and veins. Therefore, blood flows more slowly through capillaries. Slower blood velocity in the capillaries allows more time for the exchange of materials. Arteries and veins have lower total cross-sectional areas, so blood velocity is greater. Blood velocity slows as blood travels away from the heart and increases as blood leaves capillaries and returns to the heart.